All right, so this is Unit 2, Concept 3 on the cell cycle and cancer. Congrats, we've made it.
Zuzi on the track. The DNA starts to
All right, so the overarching guiding question of this concept is going to be about how does this happen? How do we start out? How do we start out our life as this fetus growing in our mother's tummy and become this, a baby, and then eventually become this, a full human being, an adult human being? How do we start out as a single cell and eventually divide and grow enough to be the multicellular organisms that we are today? Okay, and so to start answering that question, we need to talk about this. So it all starts with the fertilized egg. All right, so here is a female egg which contains DNA from mom. So here are 2 chromosomes, 2 pieces of DNA. And then the second piece is a sperm cell, which contains DNA from our dad. So there is the DNA from dad. And together, when we combine the female egg with the male sperm, that DNA from both parents, we end up with something called a zygote. All right, so once the sperm meets the egg and becomes fertilized, that fertilized egg, now called a zygote, begins going through cell division repeatedly. So as soon as those 2 are joined together, that cell is going to start dividing, all right? Because obviously we have millions of cells now. At this stage, we only have 1. So it has no time to waste. It's going to start dividing right away. All right, important to note at this stage though, at the very early stages, cell division gives rise to only identical cells. So at the very beginning, very beginning, when cells divide, they are giving rise to identical cells, all right? And don't get me wrong, when we undergo mitosis, we are duplicating a cell exactly the same. It's like having a twin brother or sister, all right? They are identical twins, okay? All right, and so I'm just showing you this image of embryonic development. So how we begin as a fertilized egg, and these are the very early stages, okay? So cell cleavage is a process by which the number of cells in a developing embryo is multiplied through cell division, all right? So we begin as this single fertilized egg, and that egg will divide and split into 2 cells. And so this would be our 2-cell stage. In turn, after that, those 2 cells would again divide into another 2 cells, lending us to a 4-cell stage. I did this earlier. 4-cell stage. All right, and so on and so forth to the 8-cell stage. And then we start eventually, now the dividing is just getting crazy, and we start turning into what we call a morula, a blastula, an early gastrula, and a gastrula. And don't get too caught up. I'm not gonna have you regurgitate this chart. Don't memorize this diagram for me, okay? I just want you to understand what's happening, that we start from this beginning, the single cell, that single fertilized egg, and through mitosis, we eventually multiply that number of cells, all right? And we turn into these are basically essentially just stages of our embryonic development. So this brings us to the cell's life cycle. So the cell cycle is a repeated pattern of growth, DNA duplication, and cell division that occurs in eukaryotic cells, which, if we remember from Unit 1, are animals, plant, and fungi. All right, and so there are 2 purposes of the cell's life cycle, which are growth and repair. So we want to grow, continuously grow, and repair any damaged or cells that we need to get rid of. All right, and so the cell cycle itself consists of 3 main phases, okay? The first phase is called interphase, and this is a period of cell growth, as well as DNA duplication. So 78% of the time, at any given time, our cells are in interphase, okay? Just a period of cell growth. All right, and so DNA duplication also happens here towards the end of interphase, and it is right before the beginning of mitosis. So it's prepping a cell for cell division. The mitosis, the stage mitosis, kind of gets all the glory, but mitosis actually has nothing to do with the separation with the division of a cell into two. It actually only has to do with the separation of that DNA. So we have our instructions, our DNA instructions, and if we're going to make a new cell, we're going to need a new copy of that DNA, right? And so the way that DNA is duplicated in a way where they're so close together, they have to be separated in order to actually split off into two distinct separate cells. So the DNA has to separate to opposite ends of the cell for that cell to be able to then close and end up as two complete standalone cells. Okay, then the actual process of cell division is in a stage called cytokinesis. It literally means cyto means cell, kinesis means movement, cell movement. And so it begins at the end of mitosis.